everyone. Good morning to everyone in the North American time zone. Good afternoon to all of you in the European time zone, and good evening to everyone else. In fact, it might be a different time of day if you happen to be watching and listening on demand. So it's my pleasure to introduce our speaker today, Mike Hildebrand. It was brought to our attention by a collaborator who recommended a paper that he recently published, and I think you'll be hearing a lot about today. But first, I'd like to do some introductions. So my name is Chris Mathis. I'm Chief Commercial Officer of AnaBios. If you have not heard of AnaBios, we're a unique contract research organization, a biotech company based in San Diego, California. We procure human tissue samples from ethically consented donors and use the tissue or cells to perform physiology assays in which we test preclinical compounds for drug discovery projects. We have essentially redefined first in human. So if you'd like more information about this unique translational company, check out our website at anabios.com. So it's my pleasure to introduce our speaker today. His name is Dr. Mike Hildebrand. He completed his PhD in cellular neuroscience in Terry Snutch's lab at the University of British Columbia. And that's when I had the pleasure of first meeting him at that time. Using patch clamp electrophysiological techniques, he characterized how the regulation of voltage-gated calcium channels impacts neuronal excitability. Following this, Dr. Hildebrand pursued an industrial R&D fellowship at Zalakis Pharmaceuticals in Vancouver, where he developed an ex vivo rodent spinal cord recording assay for Zalakis' preclinical pain research program. To further expand his expertise in spinal pain processing, Dr. Hildebrand then completed a postdoctoral fellowship in Mike Salter's lab at SickKids Hospital in Toronto. In the Salter lab, Dr. Hildebrand investigated how the differential expression and regulation of synaptic NMDA receptors contributes to both physiological and pathological mechanisms of pain. Dr. Hildebrand is now leading his own pain research program as an associate professor at Carleton University in Canada and affiliate investigator at the Ottawa Hospital, where his team is studying both acute and chronic pain processing using animal and human tissue models. So Mike, really excited to have you today. Thanks for joining us, and take it away.
Excellent. Thanks so much, Chris, and to AnaBios for the opportunity to speak with you all today across the globe. Okay, so as Chris said, my lab is investigating the pathological mechanisms that dysregulate excitability in the spinal cord to cause chronic pain. And to explore these spinal pain mechanisms, we use this translational approach where, sorry, I forgot to ask, can you see my mouse? I don't know if I'm going to assume that you can see my mouse. I forgot to ask because I'm pretty sure you can see my screen. Yep. Okay, so
yes, we can. We can, Mike. You're all set.
Yeah, thanks, Chris. I forgot to ask. So yes, so for this translational approach, we're applying findings from a rodent, our typical rodent models of pain processing, but then we're applying those findings to novel human tissue models of pain processing, which I'll talk about more in a bit. And so the ultimate goal of this research is then to identify new molecular targets for the potential treatment of pain. And so why is this so important to potentially develop new treatments for pain? And so I'm sure you are all kind of aware of this because it's been highlighted. Oh, page down, sorry. There we go. But chronic pain is a major health crisis today. We need better treatments for chronic pain. And so that's because approximately 1 in 5 people experience chronic pain, so it's highly prevalent across the globe. And besides being associated with many diseases ranging from cardiac disease, cancer, diabetes, many different diseases that have chronic pain, there can be changes in the pain pathway itself so that chronic pain can become a disease in itself, irrespective of the initial triggers that cause the initial symptoms to appear. And so this obviously has debilitating personal costs, and that then leads to huge economic costs on the order of billions of dollars because of lost worker productivity and indirect healthcare costs, and as well as those lost worker productivity costs. And so why is that? Well, one of the main reasons is because we just lack the proper tool belt to effectively address chronic pain. There's a lack of safe and effective treatments for chronic pain, and I think the opioid crisis is really illustrating that need for safe treatments for pain that are both safe and effective. And so if we want to develop new treatments, we need to identify what's going wrong at the molecular and cellular level to drive these chronic pain states in humans. And so let's look at the pain pathway then briefly. And so if we look at the pain pathway, we have pain sensors, detectors throughout our body, in our skin, on our organs that detect potential sources of potential tissue damage. And so once these stimuli activate these receptors, action potentials are transmitted along these peripheral pain sensory neurons into the spinal cord. The spinal cord then relays these signals up to the brain, which ultimately encodes this unpleasant sensory and emotional response that we know of as pain. And so when we're looking at a pathway like this, it can be tempted to think of the spinal cord as being the relay, the telephone lines that connect the receptors that are in the periphery that are responding and detecting this potential tissue damage or this ongoing tissue damage to the brain, where you ultimately have the encoding of pain. But if we're going to use an analogy like this, the spinal cord would probably look something more like that. The superficial, the outer layers of the spinal cord dorsal horn contain this complex neuronal network. And so the output neurons from this network that are found in the outer layer of the superficial dorsal horn in lamina 1, yes, they do receive those synaptic inputs from those primary pain sensory neurons that are actually detecting the stimuli, but they also receive a lot of synaptic input from excitatory and inhibitory local interneurons within the spinal cord dorsal horn, as well as descending modulatory inputs from the brain. And so it's only after integrating these various synaptic inputs that then the output neurons, the projection neurons, will send the signals up to the brain where we have then the encoding of the pain response. And so because there's so much signaling and integration that's occurring at the level of the spinal cord, you can see how if you lose this proper signaling, if you lose the balance of excitability in this network, that could lead to chronic pain. So then let's think of these output neurons in this network as a car barreling down your local highway or freeway or road. And so the driver of this car is doing what we all learned not to do in driver school. They have one foot on the brake pedal, one foot on the gas pedal. So how can the car then speed up? Well, the car could speed up by taking your foot off the brakes, so less inhibition if the car is the neuron, or pushing the foot further down on the gas, more excitation. And so my lab is exploring at a molecular level these type of mechanisms. So these changes in synaptic and neuronal inhibition and excitation that can lead to this loss of balance on dorsal horn spinal cord pain signaling and then ultimately can cause chronic pain. And so this is a validated clinical approach in the fact that if we look at this first pain synapse between these peripheral pain sensory neurons and the spinal cord dorsal horn pain signaling neurons. And so you don't need to know all the details of this diagram, but this is just to illustrate that many of the therapeutics that are currently being used to manage chronic pain are either directly or indirectly acting on various receptors and ion channels and signaling elements that are linked to excitability at this synapse. And so actually when I kind of first got specifically into this pain field as an industrial postdoc at Zalakis Pharmaceuticals, what I was surprised at is some of the basic questions on these receptors and ion channels haven't been answered compared to the brain where maybe more is being characterized. So the function of many of these receptors and ion channels haven't been kind of systematically explored, and even less is known on how these receptors and channels are dysregulated to cause chronic pain. And so that kind of forms the basis for one of the first driving questions of my research team then is, the first question then is we're trying to identify these molecular determinants. So the various types of receptors and ion channels and signaling elements that can cause hyperexcitability and then ultimately lead to chronic pain. And to do this, we use rodent models of chronic pain. Now, I'll get into that in a bit. But I guess the big question as we're endeavoring on this is, what do we do with that information? As we identify these specific molecular targets, what's next? And so for the majority of biomedical research, including in pain, if we're thinking of new therapeutics, we identify a target, we test whether blocking that target with some kind of chemical compound or activating that target relieves symptoms or treats disease symptoms in rodent models. And then typically those compounds then are tested to make sure they're in terms of they're safe, toxicology is done on them, but then there's typically this large jump to making sure they're safe in humans, and then testing whether they reverse specific disease symptoms within those human states. And so there's this huge translational divide where we identify targets in rodents, and then we test whether they're safe and whether they work in humans. And so as we all know, and I'm sure a lot of the audience knows better than me, there's the track record of this, there's often a lot of failures. And so very few of these guys are making it across the divide. And so one of the reasons could be that the underlying assumption that the pathological mechanisms and the targets we're identifying in rodent tissue, are they actually conserved? Do they have the same functions within the human state? And so that leads to the second question that my team's been tackling and trying to address is, how do we bridge this gap between these rodent findings and potential new therapies for humans? And in the second part of the talk, I'll talk about work that we've been lucky enough to be a part of, where we're starting to attempt to bridge this translational divide. But first, we need to kind of focus then on that bedrock of the basic mechanisms that we have identified in these rodent models of pain. And so one of the targets and the molecules that we're looking at are excitatory glutamate receptors, specifically the NMDA receptors. And so we're focused on those because NMDA receptors are known to be critical for driving excitability and plasticity throughout the nervous system, including at pain processing dorsal horn synapses. And that's partly because when they're activated by glutamate, they let in positive charge, which causes local depolarization, and obviously then that's involved in direct electrical signaling from one neuron to the next. But since they're calcium permeable as well, they also let in calcium into the synapses, and that activates a variety of signaling pathways that can affect synaptic plasticity and overall excitability within pathways, including the pain pathway. And so these NMDA receptors typically contain 2 gluN1 subunits. So that's one gene that encodes that, but then there's 4 different genes that can encode these gluN2 subunits. So because there's different varieties in this gluN2 subunit, that can control and create diversity in NMDA receptor functions in terms of the rate that they close, the rate that they deactivate, and also what regulators can increase their activity. And so that's critical for understanding any synapse, which specific gluN2s mediate NMDA receptors or compose NMDA receptors at those synapses. And so we wanted to address that at these pain processing dorsal horn synapses, which gluN2 NMDA receptors were mediating glutamatergic synaptic transmission at these synapses. And so to do this, we use a preparation where we dissect out lumbar spinal cord tissue from adult rats. And the adult part is critical because if we want to down the road look at models of chronic pain, we need to use the adult state where the mechanisms are fully present in the adult rats. So we dissect out the spinal cord, we section it in the parasagittal plane, lengthwise plane, because the pain processing neurons that we're looking at in the dorsal horn have dendrites that extend within that plane. We can then put it under a microscope, visualize directly the pain processing neurons that are found within, for example, here within lamina 1 of the dorsal horn. And then we can do patch clamp recordings where we can record the electrical properties of individual neurons. And so in this case, because we're looking at synaptic NMDA receptors, we can use an approach where we can control the membrane potential of the neuron and analyze synaptic responses from the spontaneous release that occurs of neurotransmitter of glutamate that then will activate these postsynaptic receptors. So that's called miniature excitatory postsynaptic currents. And so at negative membrane potentials, NMDA receptors are primarily blocked by magnesium. So these MEPSCs are mainly AMPA mediated. Whereas if we then artificially hold the neuron at really depolarized potentials, such as +60, we still have that fast AMPA-mediated component of the synaptic responses, but now we also have a more prolonged NMDA receptor-mediated component. And so the reason I'm explaining this is because a lot of our data is going to look like this. We're going to see this curve where most of the response is going to be mediated by synaptic NMDA receptors. And so we use this recording technique in combination with specific pharmacological tools. And long story short, we've identified that at these dorsal horn synapses, the subtypes of NMDA receptors that mediate excitatory neurotransmission are diverge from the typical brain synapse. At the typical brain synapse, at least in the mature nervous system, they're primarily mediated by these fast GluN2A mediated NMDA receptors. There is also GluN2B as well, but primarily it's GluN2A at a typical synapse at least. Whereas within the mature spinal cord, what we've observed is that the synaptic NMDA receptor responses are primarily mediated by GluN2B and GluN2D containing receptors, which are a lot slower and they have different regulators. So that could underpin the propensity for synaptic amplification, plasticity, and potentially as well hyperexcitability within these pain processing networks. So then the question was, how are these? There's definitely evidence to indicate synaptic NMDA receptors are critical for pain processing, including in chronic pain. So how are these synaptic NMDA receptors dysregulated in the chronic pain states? And so to investigate that, we looked at rodent in vivo models of chronic pain. So one of the example models that we use is a peripheral nerve injury model where you put a cuff on the sciatic nerve, and that causes persistent pain hypersensitivity that is mediated by changes, partly at least, by changes in excitability within the spinal cord dorsal horn. We also use a model where we inject a pro-inflammatory substance, complete Freud's adjuvant, into the hindpaw, and that induces local inflammation and swelling, but a form of pain that's persistent, and that also involves changes within the spinal pain signaling network as well. And so we test these by using von Frey, for example, mechanical testing to look at the paw withdrawal thresholds and how that changes to confirm that there is behavioral hypersensitivity in these models. And so using these distinct models of chronic pain, we then could combine them with our recordings on synaptic NMDA receptor responses. And what we found, and here's an example from the peripheral nerve injury, so the neuropathic pain model, is that there's this robust increase in synaptic NMDA receptor responses, shown here by the red line, compared to lamina 1 neurons from naive rats. So there's this potentiation of synaptic NMDA receptor responses that's specific, in that at least at baseline, we don't have a potentiation of synaptic AMPA receptor responses. So we have the specific potentiation of NMDA responses in this nerve injured state. We also used the CFA model of inflammatory pain. And we observed, first of all, that we confirmed here that in these animals, yes, there is pain hypersensitivity. After CFA injection, the amount of force or the stimulation of the paw, the threshold that requires a withdrawal, is significantly less in the CFA animals compared to baseline. And so in these animals, recordings from lamina 1 neurons shows that there's this robust potentiation. So in inflammatory pain, neuropathic pain models, we see this robust potentiation of synaptic NMDA receptors. So the next question is, well, what's the pathway? What's the upstream mechanism that's driving this potentiation of excitatory synaptic NMDA receptor responses? And so work by Mike Salter's lab and Yves de Koninck's lab had previously identified BDNF as a substance that can be released from immune cells, microglia. In other models, it can also be released from primary afferents, and it can drive hypersensitivity, pain hypersensitivity, and hyperexcitability in the spinal cord. And so one of the approaches that Mike and Yves used to investigate these roles for BDNF was they took spinal cord out of healthy rats, and they pre-treated this viable spinal cord tissue with BDNF, and then could, for a prolonged time, like an hour or so, and then they could look at what that does. And so we used the same ex vivo BDNF model and to test whether BDNF could drive the potentiation of NMDA receptors. And what we found was that, yes, there was a robust potentiation of synaptic NMDA receptors by BDNF, shown here in the red. recombinant BDNF alone, and if you block the receptor for BDNF, TREC-B, with the function blocking antibody, you could prevent that potentiation of NMDA receptors. So that showed that BDNF was sufficient to mediate this potentiation of NMDA receptors. We also did various experiments to show in these in vivo pain models, in the peripheral nerve injury and inflammatory pain models, that BDNF was involved, that it was necessary for the potentiation of those receptors. And so we've continued to work and identifying. So now we have a potential driver of this potentiation of NMDA receptors. What's the pathway between BDNF and the potentiation of synaptic NMDA receptors in these chronic pain models? And I'm just going to quickly walk you through it because it's not the focus of the talk today per se, but what we've identified, and I'm just showing a subset of the evidence for this, is that in these chronic pain states, such as in the CFA model of inflammatory pain, we have BDNF acting on its receptor within these lamina 1 spinal cord pain processing neurons to cause a downregulation of this chloride cotransporter, KCC2. So when you decrease the activity of KCC2, you lose the proper regulation of intracellular chloride. So you have an accumulation of intracellular chloride, which causes loss of proper GABA-mediated inhibition or disinhibition. So this loss of GABA-mediated inhibition or disinhibition subsequently causes a downregulation of this tyrosine phosphatase, STEP61. And so STEP61 is a phosphatase that is normally a brake on the phosphorylation and potentiation of gluN2B NMDA receptors by Fin kinase. So it dephosphorylates this kinase Fin, as well as dephosphorylates gluN2B. And so when you lose this molecular brake, STEP61, what now happens in the pathway is that BDNF causes this increase in activated Fin or phosphorylated Fin, this kinase within these lamina 1 synapses, which then causes an increase in phosphorylated gluN2B, that's one of the NMDA receptor subunits, as well as total gluN2B, specifically at superficial dorsal horn synapses. And so what this causes then is the subsequent potentiation of the synaptic NMDA receptor responses in these models, in the CFA models and in the nerve injury models of pain. And so we've identified, if we look at this pathway, what you can see is there's many different potential molecular targets that we could think about targeting to reverse this hypersensitivity. And we've shown by blocking some of these in our rodent models, we can reverse pain hypersensitivity. But as I kind of set up before, so here's a super overwhelming slide that shows all the targets and the various types of pharmacological tools we could potentially use to directly target these various molecules. But the question is, what do we do with this information? If we were thinking of potentially applying this to humans, are these mechanisms conserved within pathological pain states in humans? And so once again, we're at this divide. We're at this divide between our basic science rodent findings and potential new therapies for humans. And so we've been lucky enough to be able to start to think about addressing at least that one question of whether these mechanisms from the rodent models are conserved within pain processing circuits within the human spinal cord. And this is because we've been able to partner with a neurosurgeon at the Ottawa Hospital Research Institute, Dr. Yves Tsai. And so Dr. Tsai has this translational research program already in place where she's able to collect human spinal cord tissue from consenting organ donors. And then she uses some of this tissue to look at stem cells and with applications relating to spinal cord injury. But we've been partnering with her to look at pain processing mechanisms within this human spinal cord tissue. And I should say up front that we treat this gift of human tissue very seriously. It's a selfless act of donation from the organ donor themselves, as well as the family. And so we really try to treat it with the utmost respect and really try to maximize the amount of scientific questions that we can address to potentially lead to, hopefully one day, better treatments for pain. So what do we do with this tissue? Well, so the body from these brain dead or neurologic determination of death donors, and so I should point out these donors are typically, in terms of pain, they're healthy, quote unquote healthy, most of them at least. They don't have chronic pain. And they typically die from hematomas or brain hemorrhages. And so when the body is in the OR, it is cooled, It's perfused with protective solution to preserve the organs, including the spinal cord. And then after aortic cross-clamping, some of the organs that haven't been already removed are removed for donation. The vertebral column is opened, and then we are able, the neurosurgeon, Dr. Yves Tsai, and actually my PhD student who has been assisting with this, and I'll talk about Amory in a second, are able to actually remove and collect this spinal cord. And so the human spinal cord is immediately placed in chilled and oxygenated neuroprotective solution, which preserves viability. So we're collecting tissue within 1 to 3 hours of the heart stopping of aortic cross-clamping. And so you can see that's very different from a lot of human tissue studies where you're collecting tissue from autopsy patients so that could be 12 hours, even more after death. So the tissue here is highly viable. And so because it's highly viable, it means we can use some of the same approaches that we've been using in our rodent models of pain processing. So for example, we can take one piece of spinal cord and treat it in control conditions, but then take an adjacent region of the spinal cord and treat it in BDNF. So we can have this ex vivo model of pain processing that's directly analogous to what we've been doing in rodents. And then we can investigate the downstream mechanisms of spinal pain processing using a combination of approaches, including electrophysiology to look at the excitability of individual dorsal horn neurons. So patch clamp electrophysiology. We can do immunostaining to look at the various different types of proteins that are present in the dorsal horn and how they potentially change in these models, that including this BDNF model of pathological pain. And we can also use Western blotting approaches where we collect synaptosomes, so specifically synaptic proteins from the superficial dorsal horn, and then we can analyze the total amounts of those different synaptic proteins in the vehicle treated or BDNF treated states to see if there's changes in these proteins. And so in terms of viability, so a big question when we started is how viable is the tissue? Are there preserved synaptic synapses between afferents and between these dorsal horn neurons, and can we see these synaptic glutamate receptor responses if we try patch clamp recording? And so I'm lucky enough to have a very talented and persistent PhD student, Amory Dedick, who, as I said, is actually in the OR and has helping refining the collection of the human tissue. And then along with me, she's been performing, but I should say mostly her, has been performing the patch clamp recordings on these human neurons. And so we can see here is that we are able to get synaptic miniature excitatory postsynaptic currents recorded from these neurons. And you see here, if we look at the rate of decay of the synaptic responses from humans compared to rats, the rates are very similar, which implies maybe the underlying subtypes of NMDA receptors that are present at these synapses are conserved between humans and rats. Whereas if we look at the total charge transfer, which would be a readout of the number of receptors that are at an individual synapse in these human neurons, we see that there's a significantly greater charge transfer. So we've identified that some elements may be conserved between rats and humans and other elements may not be. And just as an example to show the viability of these neurons, in one neuron that Amory was able to record from, she was able to do, the recording lasted for over an hour and a half. So she was able to actually do pharmacology on this individual neuron and showed that a blocker of the GLUN2A subtype of NMDA receptor had no effect, which is similar to what we see in rodents, whereas blocking the GLUN2B subtype of NMDA receptor had a dramatic effect on the NMDA-mediated synaptic responses, implying a large fraction of the current is mediated by GLUN2B. And if you see what's remaining, it's interesting too, is this looks like a very slow decaying component, which would provide at least some preliminary evidence to suggest GLUN2D is also present at these adult human lamina 1 synapses. And so here you can see the kinetics of the B specific component, which has decay kinetics that are similar to what you'd expect and hypothesize for GLUN2B containing receptors. So the next question is, what about this BDNF model, ex vivo, that we've established in rodents? Do we see similar mechanisms occurring within the human dorsal horn? And so we've used this BDNF model from the isolated human tissue and shown that yes, as we see at rat synapses, BDNF triggers this downregulation of synaptic KCC2, it triggers a downregulation in active and total step 61. That was that phosphatase, that's the molecular brake. So we have the same loss of synaptic inhibition, and then that drives this increase in activated or phosphorylated fin at these superficial dorsal horn synapses. It causes an increase in phosphorylated gluN2B at these synapses, which is specific in that we don't see effects in total or phosphorylated gluN2A at these human dorsal horn synapses. And so a lot of that work was done by Jean Jouze in Paul Lambroso's lab at Yale. And so we have collaborators, Yves de Koninck at Laval University and his team, Louis-Etienne Lorenzo and Antoine Godin, that looked at the KCC2 element to look specifically at individuals so we could visualize these dorsal horn neurons, human dorsal horn neurons, with and without treatment of BDNF. And what their team, Yves' team, found was that BDNF triggered this decrease in membrane KCC2 and an increase in intracellular KCC2. So it seems that there's internalization of KCC2 within superficial dorsal horn neurons if after pre-treatment with BDNF. So then from all of these findings together, we have a working model of at least one mechanism that, and I should say most of this evidence is from males, but we have this working model of a mechanism where BDNF activates TrkB to cause this downregulation of KCC2 and disinhibition. So that's kind of loss of brakes mechanism, losing inhibition within these superficial dorsal horn neurons. That causes this disinhibition, which then we found surprisingly directly couples through step 61 to this facilitated excitation pathway, this foot on the gas mechanism where you have increased excitatory synaptic NMDA mediated pathways. And actually there's evidence, not from my team, but from Yves' team and others, that this pathway, this increased NMDA receptor pathway, actually feeds back to the disinhibition pathway. So it's this vicious loop of hyperexcitability within the superficial dorsal horn that may be driving hyperexcitability in the spinal cord, but ultimately pain hypersensitivity, based on evidence from rodent models, but now at least human ex vivo tissue models of pathological pain processing. So lastly, I'd like to just thank everyone that's been involved in this work. So from my lab, Anne-Marie's done a lot of the work in rodents and human tissue. Chaya was critical as a master's student that was critical for establishing that human BDNF tissue model of pain. Hadir has been looking at the different subtypes of NMDA receptors. She's a master's student as well. And then collaborators, including Yves Tsai at the Ottawa Hospital Research Institute, Mike Salter, where I did my postdoc and started on these investigating these mechanisms at Sick Kids and his team, Paul Lambroso at Yale, and Yves de Koninck looking at KCC2 and those mechanisms at Laval University. And obviously all of the funding that makes all of this work possible. So thank you all. I really appreciate you tuning in, and I'd love to hear from questions. Thank you.
Thanks, Mike. That was great. I really appreciated you presenting your groundbreaking work, and it's interesting. It doesn't always happen where you get the translation like that. So that was a very important work. So we do have time for questions. So there's two ways to ask questions. You can write them in the chat, and Mike and I will see them, and you can answer them. Or you can, if you have audio capability, you should be able to raise your hand or whatnot, and we will let you ask as if you're at a conference. And while the questions start coming in, I have a question for you, Mike. So I love the slide at the beginning where you showed the complexity of the wires, you know, as a relay station, and the complexity of the spinal cord itself. Yeah. How do you deal with interactions? You know, it is kind of in the middle. How do you deal with interactions from dorsal root ganglia and the brain? Because you're getting input from both of those. Do you have any idea? Have you addressed that level of complexity, or have you just focused on the molecular targets and the spinal cord?
So that's a good point in terms of these synaptic inputs are coming from different sources, and how do we look? So a lot of our work would be just looking so far mainly at just overall synaptic responses within the output neurons, within the dorsal horn neurons. But we have done some work where with Mike's lab, Graham Pitcher, for example, has looked at afferent evoked specific responses in these dorsal horn neurons and shown at least the same general subtypes of NMDA receptors are involved in that. But that is a next level of the type of questions we want to look at is, for example, different subtypes of dorsal horn neurons, and then these different inputs. And I should say the preparation as well with the slice, at least directly, we're more looking at it in isolation. We don't have intact DRGs with our preparation. We don't have intact descending modulation from the brain because we're looking at tissue slices. Whereas in the pain models, obviously in our in vivo models, all of that would be intact.
Thanks, Mike. Yeah, that's definitely a dream of ours at AnaBios, is to be able to develop a preparation where we have DRG neurons connecting to spinal neurons and so forth. But one step at a time. So the questions are starting to come in. I'll read them. First question is, what is the molecular mechanism that links the decrease in chloride gradient and lowers levels of step 61?
Oh, that's a great question. We haven't, so we're thinking on that. So we think at least upstream, we've gone back one further, and my guess is that PKA is involved in the step 61 involved in the dysregulation of step 61. And so we have, but we haven't directly tested that yet. So we think it's calcium dependent. There's evidence to suggest it's calcium dependent as well. And so it could be part of that, because remember, I did say there is evidence to show that it's a circular loop where there's NMDA mediated activity involved with that. So there could be an NMDA component itself. And that's where it becomes very challenging, is that if NMDA is the target of the potentiation, but also involved in the potentiation, it gets to tease out some of these mechanisms, it gets challenging at some point. So we have to really design our experiments carefully to address this. But thank you for that question.
All right, the next question is, we got a couple more questions online, and then someone's raised their hand. So if that person could hang on just a moment. So why don't NR2B blockers work in humans? Many pharma companies have tried this.
There's many, it could be a lot of factors. Obviously, there's off-target effects as well. So it could be the dose that's required to affect the pain processing is affecting many processes in the brain. So that could be one of the reasons. There could be other drug-related reasons in terms of how it's metabolized, those kind of issues as well, how it's targeting. So I don't have as much expertise in specific clinical things, but what I could say is that's where D, for example, might hold promise in that it's not highly expressed at mature synapses in the brain, whereas we do see it seems to have this prominent role within these circuits. So that could be one element that maybe could hold some promise for as a target.
Yeah. Okay. And the next question is, have you tested whether prolonged Gaba inhibition in your model reverses the NMDA response in the BDNF model?
We haven't done Gaba specifically. There is other groups that have done the reverse, that have shown prolonged Gaba stimulation. Gaba inhibition would trigger the pathway. So there's one showing bicuculline, for example, can trigger this pathway. We have looked at the effects of blocking disinhibition with acetazolamide, which blocks I won't go into how it acts, but it blocks the effects of disinhibition, and shown that yes, it does reverse our specific mechanism, and it reverses pain hypersensitivity.
Okay, thanks. And I believe that Simon Shea has a question. If he could be unmuted, go ahead, Simon. Oh, looks like he sent in the question. Are these studies reinforced?
Mike? Yeah.
Hi, Simon.
Yeah, hi.
Thank you. Do you want to ask the question? Do you want to ask the question?
Oh, sure. Yeah, thank you so much. I really impressed by the studies. My naive question said, for most lab, we still have to start with a rodent.
Absolutely.
From your study, it seems look like to early stage that the rat model or rat tissue still could be mimic or similar to a human tissue you compare with. I mean, I agree. Ultimately, we need to using a human tissue to directly provide evidence. But I just very general question to you.
Yeah, that's a great.
Yeah.
That's a great point. Absolutely. Like we need to start, and we can't, obviously we have limited access to tissue. We can't address all of the questions. So we need these rodent models. And yes, what we're seeing is that at least for these mechanisms, at least in this model, the BDNF, and I should stress it's a model of pathological pain processing, but we do see the same mechanism in the BDNF model as our in vivo models. But yes, it looks like that the mechanism is conserved from rats to humans in males. We have ongoing work and actually that was partly from working with humans that's helped shed light on differences in males and females. And so we're currently investigating how it might diverge, maybe not across species, but across sex for some of these pathways.
Mike, there's another question for you. KCC2 and NKCC1 are co-regulated. Do you know what happens to NKCC1 activity in your model?
It doesn't change in these. So I haven't done that work, but Yves DeConning's labs looked at it, and the effects are specific to KCC2 from what I remember in terms of these mechanisms.
Okay. And then another question, and for those of you still on the line, we have time for questions, so feel free to send them in. Are the mechanisms involved in regulating GLU-N2B activity so selectively expressed in the spinal cord?
So there are some of these mechanisms. I think their impact could be different in terms of, because the relative role, for example, of GLU-N2B at these spinal synapses, but there's evidence that some areas of the brain, at least some elements of this, are conserved. And so they could be related to some elements to plasticity and learning and memory, but I don't think all elements. And I should stress, within the spinal cord itself, we've seen this pathway is specific to the superficial dorsal horn. We do not see the same upregulation, downregulation of our targets in the rest of the spinal cord tissue. And actually, even if we just take whole tissue homogenates, we don't see the effect. So it's specific to synaptic proteins within the superficial dorsal horn.
Okay, thank you. And we have another person who's raised the hand. Sasha Alice, go ahead.
Hi there, can you hear me?
Yeah, can.
Yeah.
Hi, Mike. Great talk. Just wondering if you ever get spinal cord from chronic pain patients.
We have, so the tricky thing is we don't have a full, like these are organ donors. So you get some, we have access to the available medical information, but we don't have a full profile. So we can kind of infer sometimes, for example, that, and so ones we've excluded from their study is, for example, a patient that has been on opioids. So we'd exclude that. So we do have those patients, but the tricky thing is the low end for those and the lack of the really detailed medical history. It makes it a little challenging, but we do have a bit of preliminary evidence to suggest there might be things that are different between them, but it's just not enough to make firm conclusions, at least at this point. But that could change as we get more donors.
I would add that at AnaBios, we do have a very extensive network in the U.S., and we procure spinal cords from ethically consented donors. And we do get spinal cord samples from chronic pain donors. So those samples can be used for gene expression studies and whatnot. And we're also developing a physiology assay.
Yeah.
So another question for you, Mike, is, thanks, Sasha. Some analgesic work in rodents, but failed in human clinical trials. Have you identified any elements that rodent pain, how they're different from humans?
So
rodent pain is different?
Yeah, that's a great question. These are all great questions. So we're still pretty early in this process, I'd say, in terms of investigating the physiology and the pathology in these tissue. But the one thing I'd say is the synaptic responses, there look like there are some differences where we're continuing to investigate. So the relative contribution of the different subtypes of NMDA receptors might be different in the superficial dorsal horn of humans compared to rats and rodents. So that's one area we're following up. So for example, the relative contribution of GLU-N2D and even B might be different in humans versus rats. So we're continuing to follow up on that. And many, like as you can probably imagine, there's an infinite number of questions that with approaches such as this that need to be investigated. So we're just scratching the surface. And I would hypothesize definitely, if there's difference between mice and rats, which we've seen in pain, I would definitely hypothesize that there's going to be some differences from rats to humans.
Mike, I have another question for you. So it was interesting to see the difference in current amplitude between the rats and the human. Is that just the size of the cells? I don't remember if that was normalized.
Yeah, it's because, and that's obviously intuitive. Actually, even when I was doing the recording, you think that, but remember, we're only recording individual synaptic responses. We average So responses from many synapses,
okay.
it's individual. So it's not the size of the cell because they're individual synapses. That being said, maybe it's the size of the synapses. Maybe there's more room, they're bigger, there's more receptors. It could be receptor density. There's various factors that could drive that. But no, it's not the overall size of the cell.
Okay. And do you think that there's any significance to that?
Well, it shows there's a robust. So we need to look at the AMPA, like the ratio of NMDA to AMPA, and we haven't done that. So that's another follow-up, because it could mean that maybe there's the ratio of NMDA to AMPA, at least at these synapses, is different. And so that would have huge, if NMDA receptors are a lot more highly expressed at these synapses, that could have big implications for understanding neurotransmission at this pain synapse.
Okay, and how about as a treat for the people that are still on the line? There's quite a few still. Tell us what's next. What's coming out of your lab next, and what can we expect at pain conferences this year?
I kind of teased it, and I'm too early to talk too much about it yet. But looking at the female to female, so we're looking at these models, these mechanisms in females. Another big thing, we're trying to leverage this model, because obviously those patch clamp recordings on human tissue are very challenging, as Anne-Marie can, I'm sure, attest to if you chat with her about it. And just so, and throughput's very low. So we want to maybe try to develop other ways to look at excitability within the spinal cord, using, for example, voltage sensitive dye imaging to look at excitability in the whole network, and to really increase the amount of information we can gain from each donor, to really try to maximize the scientific questions that we can answer with this precious tissue.
Absolutely. And one other question that came in, what is the frequency of organ donors to get spinal cord tissue?
So I
can answer it for AnaBios.
Yeah, so AnaBios, you have a network of hospitals, from what I understand, whereas we have one neurosurgeon and one hospital.
Right.
So it's highly variable. So we were averaging the first couple of years, like one a month probably. That can change, and now we've had less in the short little period. But it's highly unpredictable, which is very hard, as you can imagine, for the students involved. That means they're on call 24/7, and when the donor comes in, that's when the experiments start, which can last for up to like 18 hours or beyond. So it does make for challenging. You need very dedicated researchers involved.
Yeah, with AnaBios, with our extensive work in the U.S. that we've developed over the past 10 years, we get 2 to 3 sets per week, or 2 to 3 spinal cords per week, and so the pain donors are less frequent, so maybe 2 to 3 per month. But it's all standardized. It's quite the same every time. And just like you said, with your graduate students, our team of scientists and technicians at AnaBios are like firefighters that come in at 2 in the morning and are always available to deal with these precious samples. So. Well, Mike, thank you so much for this great presentation. I look forward to seeing you at conferences this year. And I just want to remind everyone that's still on the line, if you want to share this with friends and family, it will be available for download in the coming days. So thanks again for joining us, and have a great day. Bye, everyone.
Thank you.